Hi everyone, I'm Alexandre, a PhD student from the University of Mons, working on the PoshBee project. In this video, we'll not talk about domesticated bee species, but rather about wild bee species, which, even if you may not always see them, represent over 2,000 species in the world. These species can be very different in terms of size or colors. They can be solitary or social, they can nest in soil or in cavities, and they can even be adapted to feed on specific flowers. Their decline could lead to a risk for ecosystems functioning and coherence. Unfortunately, most of the studies on bees are conducted on honeybees or bumblebees, mostly because of the difficulty to keep wild bees alive in laboratory conditions during their whole life cycle. However, with the PoshBee project, we wanted to know more about these species and try to understand how pesticides could impact communities of wild bees, and if there was an interspecific variation of these impacts.
To do so, we started to capture as much species as possible during the whole flying season, from March to October. Here, you have an example of the capture of Andrea vaga, an oligolectic solitary bee species that feeds on willow tree and nidificates in soil. Captured individuals were then isolated in small cages, waiting for either annual or topical pesticide exposure. All species were exposed to the exact same dose of pesticide, and the mortality was monitored for 48 hours. The idea here was to look at the sensitivity of various species toward agrochemicals, and to see if some specific traits could explain this variability.
Bees can be exposed to pesticides in many different ways. They can be directly exposed during the pesticide application, or indirectly through pesticide residues that can remain in plants for weeks or sometimes even for years after the application. These residues can also be found in the soil, which may constitute another exposure route for soil nesting species.
We tried to assess how important this exposure can be. To do so, we collected eggs of Colite cedere, a soil-nesting solitary species that feeds mainly on ivy. This species lays its eggs on top of a pollen stalk inside a fine protective cell. Several cells were collected and brought back to the lab. We then put the cells into different contaminated soils. During 1 month, cells were frequently taken out of the contaminated soil, and the inside of each cell was analyzed to look for pesticide residues.
Thanks for watching the video. Don't hesitate to follow the PoshBee project on Twitter, and if you have any question, please leave a comment below. I will be happy to answer. Bye!